Already recorded. Profesor Arjo De Marco je sodelavec laboratorija za vede o okolju in življenju, hkrati pa tudi redni profesor na fakulteti za znanosti v okolju, kjer poučuje predmete, kot so biokemija, rak v okolju. Sedaj vam bom predstavil kratko predavanje in sicer z naslovom Potovanje skozi rudnik.
Today, we'll talk a little bit about diagnostic. What does it mean, diagnostic? It means that you can have a possibility.
Petra, again, we experienced the same issue. The sounds again stopped.
Anything
But
else
now?
that you are
Now it's
interested
okay.
in? So probably you have listened in this last period about necessity of detection of how much, COVID, so virus particles are present in samples from people and so on. But the same is true also if you wish to analyze how many bacteria are present, for instance, in your milk or your fruit juice and so on. Yeah. For detecting the different elements that we wish to identify in the different samples, usually we use antibodies. Why antibodies? Because this class of molecules represent the best way for being precise and selective. So it's possible to identify exactly what we wish without problem of cross-contamination with other elements that are present in the sample. And among the antibodies, that are a large class of molecules, we are working with nanobodies. Why nanobodies? Well, now I'll try to explain you with some graphs. So we say our problem is to identify something. Yeah. So this is the object we might identify, and we can consider that in the sample, this is together with a lot of other stuff. Yeah. Okay, so you can understand it can be difficult to identify just these molecules in a complex system. Okay? So what we need, we say, is a molecule that can somehow eliminate all the background and specifically bind to the target, so the antigen. And it is the antibody. Yeah? Antibody. Okay. So far, so good. Now, what is the problem with the conventional antibodies? It is a large molecule. So an antibody is as large as I draw it now. Yeah. So it means that I need all this large structure for having the active site that recognizes my antigen. So this means that it's necessary to try to simplify all this molecule to have a lower cost and a simpler way of producing. And now comes out the nanobody. In this case, I don't have any more all this stuff here, but I have only the active side. same work as the other, but can produce in a simple way. How can we get nanobodies? Well, we can use large libraries in which there are many of these nanobodies with small differences here, and this small difference enables to get something roughly different in terms of antigen. Yeah? So if I have a collection of nanobodies as large as 10 to the 9, so 1 billion of nanobodies, statistically, I'm almost sure that I will be always able to find at least one nanobody that is specific for my antigen. Okay? So we work with this kind of large collection and we select out the right nanobody. And once we have these nanobodies, we can engineer this molecule in order to get what we say from a nanobody, yeah, to a reagent. And what we need for passing from a basic research molecule to a reagent, well, usually we have to add something, a tag so-called, to the nanobody in order to enable the fusion molecule, yeah, to make the activities we require to a reagent. So for instance, yeah, if I have my initial antigen, and I have my nanobody fused to the tag enabled to visualize the antigen. Because, for instance, this part here is a fluorescent molecule. Of course, when you work with something, you wish to show the positive side of your object. There are also negative side of libraries like the one of nanobodies, where we have just discussed previously. There are some problems that can arise from the use of these reagents. So for instance, sometimes the selectivity or the affinity, so the capacity of binding is not good enough, and so on. Sometimes they are not soluble enough. Yeah? But all these parameters indeed can be improved by, for instance, in silico improvements or modification. So it means that we have informatic-based system that enable further optimization of initial binders.
Hi, I'm Claudia. I'm a young researcher in a laboratory from environmental life science, and today I want to show you a little application of my work. I work with protoplasts. Protoplasts are plant cells without cell wall, and for this reason they are very useful and they are used in several applications. For example, they are used in genetic transformation or to study embryogenesis, cell division, and several metabolic pathways. To isolate protoplasts, I'm using populous leaves, so forest plants, and here we can see that we have leaves. So we start from leaves, and then we cut leaves in little stripes using by a simple knife, And we use an enzymatic method. Indeed, to isolate protoplast, we can use an enzymatic or mechanical methods. We choose to use an enzymatic method because it's less hard and also less laborious in comparison to a mechanical method, and includes the use of two different enzymes, mastoenzyme and cellulase. They are two enzymes derived from fungi, and they are able to digest cell wall. So by means of these two enzymes, we get protoplast. So after cut leaves and stripes, we can add the digestion solution. And just to cover leaves. And after this, we just incubate our sample at 30 degrees with shaking for 6-7 hours. After 6-7 hours of incubation with our digestive solution, our leaves appears like this. So it means that they release protoplast. And after this, we filter and centrifuge this solution in order to remove as much as possible debris and other stuff. And we got this ugly solution. To detect our protoplast, we can use epifluorescence microscope. And so we just put a little drop on a slice of the solution. And in this way, we can see our protoplast. And it is possible because of fluorescence of the protoplast due to chlorophyll content. Indeed, chlorophyll absorbs the green, blue channel, and emits the red channel. So at the fluorescence, we can see this picture. These are our protoplast, and we can see this little circle, and also here we can see better our protoplast. After this, once obtained protoplast, we can start with the other analysis. And so, as I said before, genetic transformation, study of mechanism membrane, and so on. And in our case, especially, we use this protoplast to identify specific factors involved in transformation and data express on protoplast.
Hello everyone, my name is Sandra and I'm a postdoctoral researcher in the University of Gorica, and I would like to show you the purification process of recombinant protein. In this case, the recombinant protein here is actually a fluorescent one. As you can see on the plate, it's pink. So how do we get here? What we do is that we try to clone the DNA for this specific protein we want to produce. And we do that using the host cell, which is bacteria. And the bacteria we use here is the Escherichia coli, popularly known as E. coli. So we produce our recombinant protein here. Then we do that in a large quantity by introducing a colony of the bacteria on the plate into a large volume of medium, which contains all the nutrients needed for the bacteria to grow. Then upon that, we try to get the cell lysate. Then we introduce the cell lysate in the column here. So this machine is actually what we call the actor, and it is the fast performance liquid chromatography. And on this machine, we do a process called the affinity chromatography, where you have a column that contains either nickel or cobalt. which actually can bind to your E-stacked protein. So by that, you are able to purify your E-stacked protein. So the protein you get from this is actually E-stacked. So this process could actually be used because it has an E-stack fused to it. So you get your protein from this, and at the end of the day, you have your purified protein. And after this, you can actually determine the purity, the concentration, and the molecular weight of this protein using the SDS page, which is the sodium dodecyl sulfate polyacrylamide gel electrophoresis. And you can also check the concentration using like Bradford acid. So at the end of the day, you have your purified protein, which you can use for further application. Thank you very much.
So that was the video.
Well, thank you. Thank you very much. It was fine. Yeah, there was a little bit, some parts not clear, but I think we need to improve the technical these issues like the computers. Yeah. In general, it was very fine. I think. And I will stop it now.